This is an overview lecture on DNA replication. As I stated in class, most of what you're going to hear, this is a review for you. You've heard before. And so, but what we need to focus on is really the application process of this learning. The learning objectives are as follows: that at the end of this session, you'll be able to articulate how the search for the molecule of inheritance was, why DNA was an unlikely candidate to be the molecule of inheritance, articulate Griffith's, Avery, McCarty-McLeod, and Hershey and Chase experiments and how critical they were in proving to the scientific community and to the whole world that DNA is the molecule of inheritance, and then articulate the roles of Charkov, Franklin, and Watson and Crick and Linus Pauling in the discovery of the structure of DNA. This is going to be the first part of this lecture. And so, for many years, people realized, since the days of Mendel, people realized that heritage was passed on from generation to generation. They just did not know what the molecule was that was involved. They knew that it had to be a molecule that was that had the capability of being replicated and passed on from generation to generation and carried traits with it. Frederick, who was a Swiss chemist, recognized early on, and he discovered what he called the nuclein. And this was a molecule that was found in the nucleus, and he thought that it would be a good candidate as the molecule of inheritance. And this molecule had carbon, hydrogen, phosphate, oxygen, and of course nitrogen. Frederick Griffith was probably the first one that did an experiment that kind of began to lead folks on focusing on the molecule of inheritance and its ability to pass on from generation to generation. What Dr. Griffith did was he worked with Streptococcus pneumoniae, and he found that there were two different strains. One of these was smooth and the other one was rough. And the smooth one had the lipopolysaccharide capsule, while the rough looking strain did not have this capsule. He found that if he were to take the smooth colony and injected it into mice, the mouse will die. If he took the rough colony and injected it into a mouse, the mouse will live. And then he did another experiment, and that is if he were to subject the virulent or the infective smooth strain to heat and kill it, and then he would inject it into the mouse, but the mouse will survive. So whatever that factor was that was responsible for killing the mice, it was sensitive to heat. And then he did a very critical, very ingenious experiment, and that is that he would take the heat-killed smooth and mix it with living R strain, rough strain. And when he injected that mixture into mice, the mice died. And when he isolated and cultured the bacteria from the tissue of the mice that had died, he discovered that the living rough strain had been transformed or changed to the smooth strain. And so his conclusion was that the virulent substance is in the smooth strain, and that the transforming principle was passed from the smooth strain to the rough strain, and was able to transform the rough strain to become virulent like the smooth strain. And then this is where his Griffith's work stopped. But then a group of three individuals, Avery, McCarty and McLeod at Rockefeller University kind of took over and simplified that experiment that Griffith carried out. What they did was they took the smooth strain of Streptococcus pneumoniae and they subjected it to heat to kill it, and then they isolated the extract from this, and then they took the rough bacterium and they sprinkle or they mix these homogenate with the rough strain, and they saw that they could transform the rough into the smooth colony. They then took that homogenate or filtrate and subjected it to protease to remove all the proteins, and when they mixed it with the rough strain, they found that it still had the ability to transform rough into smooth. And so it was not protein. And when they mix it to ribonuclease to remove the RNA from the extract, and then they mix it with the rough bacteria, and they found that it was still able to transform the rough bacteria into smooth bacteria. So it was not RNA. But when they subjected that filtrate or homogenate to DNA, so deoxyribonuclease, to digest the DNA content of that homogenate, and they mix it to the rough bacterium, that the ability of that homogenate to transform the rough bacterium had disappeared. So the conclusion then, their final conclusion is this, that DNA is the active factor that was able to transform the rough bacteria into smooth bacteria and cause it to be virulent. So the active principle is DNA. Now, people were still very skeptical. The purity of the homogenate, they just weren't convinced until Hershey and Chase, Alfred Hershey and Martha Chase, in the early 1950s, these two were studying bacteriophage, and these were viruses that infect bacteria. And they were studying the life cycle of the phage, and they found that if they were to infect the bacteria with the virus, that after one generation, that the virus particles would be generated within the bacteria, and then bacteria will lyse and the viral particles will be released. And so they asked the following question: since we're working with viruses and viruses are very simple, what if we decided to do an experiment to prove what the transforming principle is? And they would do that by labeling the DNA with P32 and the protein with S35. Now, and then, so what they did was this: they would take E. coli and infect them with virus whose DNA was labeled with P32 or whose protein code was labeled with S35. Now, something about virus that you need to know is that this is very simple. The core inside the head is DNA, and the code outside is the protein. So you could separate the two very nicely. So they labeled the DNA with P32 and they labeled the protein with S35. And so they infected the E. coli, and at the end they pellet the bacteria and they lyse the bacteria and they began to look for the radioactivity. And so here's what they did. They did a very simple experiment, and that is that they mixed the bacteriophage with the E. coli, and then they put it in a Waring blender, and they blended at a speed. They pressed puree and they blended at a speed that will knock off the viruses, and then they would pellet the bacteria, wash it and rinse it several times, and then they would lyse the bacteria and they would identify whether or not the P32 is inside the bacteria or the S35 was inside the bacteria. And what they found was that if they were to do label the protein code with S35 and they infect the bacteria, knock off the phage with the Waring blender, and then they wash the bacteria, analyze the bacteria, and look at what's inside the bacteria. They found that S35 was not present. But what they found, on the other hand, was when they labeled the core of the bacteriophage with P32, which labels the DNA, and when they infected, knock off the phage, and they pellet the bacteria, wash, and lyse, they found that they were able to identify radioactivity within the E. coli. So this proved once and for all that the molecule of inheritance, or the transforming principle, was indeed DNA. And from this experiment, people were really convinced. Now, what is the structure of DNA? There were two very famous fellows, James Watson and Francis Crick. Watson was an ornithologist from the University of Indiana, 23 years old, was interested in finding out what the structure of this was. And Crick was a physicist at Cambridge. Now, it turns out that these two fellows talk a lot. And so James Watson left Indiana and he went to Cambridge, and he began to team up with Dr. Crick. And they had coffee and they talk a lot and they talk some more. They did very little experimentation. What they were banking on was all the information that had been amassed from the work on a lot of scientists in different countries, in different labs. And so they began to examine them. But there was a group, Dr. Wilkins' group at King's College in London, which was about 75 miles away from Cambridge. And there was a young lady by the name of Rosalind Franklin. Now, this is an X-ray crystallographer. She was working on crystallizing proteins, and she was working on crystallizing DNA as well. And she would look at the crystal structures using X-ray diffraction grading and predicting what the structure of these molecules were. Now, her interaction with Watson and Crick was not very good. He thought they were two fellows that had no business in science because they didn't do a whole lot of work, and she didn't have a whole lot of respect for them. Linus Pauling at UC Berkeley was really busy on trying to come up with the structure of DNA, and he is noted for winning the Nobel Prize in chemistry, and he also won the Nobel Peace Prize. Not very many people win multiple Nobel Prizes, but he was famous for doing that. And so he was working about the same time on trying to beat everybody to come up with the structure of DNA. And then there was Hungarian-born scientist at Columbia by the name of Charkov. Charkov was a chemist, and he was a very brilliant chemist. Now, on his way to Great Britain, he met with Watson and Crick, and he came up with the following conclusion: these guys are not very good scientists, and they have no business in science. But one day, Watson and Crick decided to propose a model for the DNA, and they released it to the public, and it was not a double helix but triple helix, and nobody accepted it. In fact, they had the nitrogen bases oriented to the outside instead of toward the inside, and no one accepted it, and Cambridge kind of banned them from ever releasing anything like that. But so they kept on working, and everybody that was working on trying to elucidate the structure of DNA continued to work. And so on one of their visits, Watson and Crick went down to King's College, and they began to visit with Rosalind Franklin, and they were trying to see the pictures that she had, her X-ray diffraction pictures that she had, and she wouldn't let them see them. She didn't think they deserved to see anything, and besides, she had concluded that they were not very good scientists. They had no business in science. And so, disappointed, Watson and Crick were walking out, and in the hallway they met Rosalind Franklin's boss, Dr. Wilkins. And Dr. Wilkins said, "Hey, listen, step into my office and I have something to show you." He whipped out a picture that Rosalind Franklin had generated from DNA, her work on DNA. And Dr. Crick, being a physicist, knew exactly what the structure should be from that picture. And so they went back and they proposed their model, the DNA double helix. And of course, it was published, I believe it was 1956, on the cover of Nature, and they were very, very popular. But Dr. Chargaff and Dr. Franklin were very bitter because they thought that these two did not deserve to get the recognition for elucidating the structure of DNA. Now, if you don't remember the basic structure of DNA, please go back and review it in any basic textbook. But DNA consists of units of nucleotides, which are phosphate, deoxyribose, and nitrogen bases. They're linked by phosphodiester bonds. There are two strands that are running anti-parallel to each other. Nitrogen bases are numbered with normal numbers 1, 2, 3, 4, 5, and 6, while the deoxyribose sugar is numbered with numbers containing prime, just to separate the two sets of numbers. And double helix has sugar and phosphate backbones, alternating sugar and phosphate backbones. Nitrogen bases are oriented toward the center, and the DNA molecule is held together by weak bonds called hydrogen bonds. And this is the structure. You have on the right the picture of the DNA double helix right here, and then right here on the left you've got the structure where you have one strand that goes from 5 prime, 3 prime, and the other strand from 5 prime or 3 prime to 5 prime. You have the deoxyribose and phosphate, alternating deoxyribose and phosphate on the backbone, and you have oriented to the center are the bases where A pairs with T with 2 hydrogen bonds, and G pairs with C with 3 hydrogen bonds. And so this is the basic structure of DNA.